As you may know, we divide classically the brain according to two streams of processing, the dorsal and the ventral stream. The dorsal stream mostly being related to spatial information, where are things, and the ventral stream being related to the conceptual semantic aspect of things. What are those things? And the way, this stream divide in the occipital, the parietal, and the temporal lobe is quite well known based on anatomical investigation and electrical recording in monkeys, but how it extends to the frontal lobe is actually quite unknown. So we were thinking, like, how about we investigate this question with the lens of neuroanatomy, starting from looking at how the posterior part of the brain speaks to the anterior part of the brain via white matter connections. And if you do so, you can see that we have three main branches of connection connecting the posterior part of the brain with the anterior part of the brain, namely parietal and frontal lobes. These branches are called superior longitudinal fasciculi, and they divide in three. The dorsal one connects the superior parietal lobule with the superior frontal gyrus in light blue or cyan. The second one connects the intraparietal sulcus and the angular gyrus with the middle frontal gyrus and frontal eye field. That's the second branch in dark blue. And the third one connects the temporal parietal junction, supramarginal gyrus, with the inferior frontal gyrus, and that will be the pink one that we can see here. Now, this is anatomy, and how anatomy relates to function is the main question that we're exploring when we're doing fundamental exploration research on the brain functioning. And so we try to assess every fMRI studies that have been reporting co-activation in the frontal and the parietal lobe over the past 12 years. We've been doing that manually in order to investigate whether we have different function associated with these different tracts. And if you do combine all these studies, I think there was like 800 and something, you can see that you have strong pattern of co-activation that happen in the brain for 14 different functions that are clustered into a main domain here. So you have the visuospatial function that includes saccades, mental imagery, voluntary oriented attention, automatically captured attention. You have working memory, whether this is verbal or spatial, language, which involve phonological processing and semantic processing, sequences and inhibition for the motor tasks, and other functions that I couldn't put in a subgroup, such as number manipulation, emotions, decision-making, and mirror neurons. All these functions co-activate the frontal and the parietal lobe. And they also activate other areas in the brain, but here the focus is on the frontoparietal cortex. And the first things that happened, we look at this and we were like, so how do these functions differ really? Because there is so much overlap in the brain regions that are activated here between very different functions. So we essentially, instead of segregating them, we tried to identify main components, main ingredients of similarities across those functions and try to decipher the mechanisms that will be supporting this similarity. And the way we do that, we applied a framework which is called the principal component analysis, which does exactly what it says. It extracts principal component from your pool of neuroimaging data. And when we did so, we extracted a lot of components, as you can see on the chart on the left, but only the two first together were the ones that represented most of the variance, representing 70% of the variance of the data set, with the remaining being like a long tail of components that we usually call residual or noisy components. Those two first components together, if we manipulate their intensity, can reproduce all the maps that you've seen before with a 70% accuracy, like exact numbers in each voxels, which is quite impressive if you think about it. And then we project back those components onto the surface of the brain. And the first component happened to be a ventral frontoparietal network involving the supramarginal gyrus, temporoparietal junction, inferior and middle frontal gyrus. And the component number two being a superior network involving the superior frontal gyrus and the superior parietal lobule, with the intersection between the two being at the level of the frontal eye field and the intraparietal sulcus. So, how do you interpret the function of these components? And to do so, you can look at how much each function we've been exploring loads onto these components. And here you have the representation of the table of the loading of each component. As you remember, component number one is the ventral pink one, and you can see that high values for this component happen to be mostly for mirror neuron semantic processing, verbal working memory, and phonological processing. On the other hand, the component number two has high value for a different pattern of function which involve voluntary oriented attention, automatically capture attention, saccades, mental imagery, motor sequences. So it looks like you have a segregation between spatial and non-spatial function, non-spatial function being ventral in this frontoparietal network, and spatial function being more dorsal in this frontoparietal network. So you might think like, hey, but that does remind me a little bit the segregation of the brain based on white matter connections, which we try to assess here. So those are like the projections of the white matter tracts just before you had the functional activation of the two components and the overlap. And you can see that visually there is a good match that we quantified here using statistics. You can see that the spatial component loads a lot onto the SLF1, and the non-spatial component loads more onto the SLF III and the SLF II, and the intersection is really dominant for the SLF II. So it looks like you're manipulating something spatial, it's happening in the SLF1, non-spatial in the SLF III, and SLF II will allow you to integrate those two domains at a very high level. So to come back to our point, if like the segregation line between the where path and the what path in the brain were located in the Sylvian fissure for you, you might rethink a little bit your mental segregation to bring it a little higher, since apparently non-spatial function will also involve the ventral parietal and the ventral frontal lobe here. Now, I've been working for a long time on white matter connections, and something that I always find fascinating is the way when you look at white matter connections, they seem to really help understand how the brain functions. And so it's, let's try to ask this question together. Are connections defining the functional specificity of brain areas? And if they do so, we can go the other way around and try to decode the brain organization by unfolding its pattern of connections. This is like a fascinating idea, which is absolutely not mine, has been defended and suggested by many fantastic authors, such as David Van Essen, who say that areas can be arranged in a well-defined hierarchy on the basis of their pattern of interconnections. Marcel Mesulam, who say nothing defines a function of a neuron more than its connection with other neurons. And Carl Ziles, who said afferent, efferent, and intrinsic connections, as well as cellular types and their properties as structural basis for a brain region function. You have some example of their work at the bottom part of the slide. On the left, you have those circuits have been drawn by David Van Essen in the 80s. And on the right, you have this fantastic method, which is polarized light imaging, that allow you to see the brain connections at a microscopic resolution. So that's great. So why don't we have an atlas of a functional brain region based on their connections? Why is it not available? Well, because those connections are not easy to assess in the living human brain. And for a long time, people have been using primates and do axonal tracing in the brain of primates in order to assess the structural anatomy of the connection of the brain, and then infer that the human brain will be the same as the brain as primates. And even though you hear a lot about the term connectome, saying like the entire whole representation of the connection of the brain, we don't have a connectome really for the macaque brain because we never been able to inject the entire macaque brain. You have the depiction here of the most advanced atlas of the connection of the macaque brain. And on the left, the black areas correspond to the patchy distribution of injections that allow to measure the connection in those brain. They do not cover the entire brain. Now, on the other hand, we have tractographies that give us like gorgeous representation of the entire human living connectome. However, this is at a resolution which is quite macroscopic. Now we can go down to 1 cubic millimeter, but 1 cubic millimeter is about 1,000 times bigger than a connection. So these streamlines that you see here are not individual connections, but group of connections. And this represents a limitation in the way we can assess all the fine circuitry of the human brain. Now that I did a disclaimer on the limitation, we can say that how about we'll try to use the information that we can derive from the structural connectivity based on diffusion weighted imaging tractography to divide the frontal lobe into subregions using a principal component analysis. So to do so, we'll try to look at how every voxel of the frontal lobe is connected to every voxel in the same hemisphere plus extra regions, and run a principal component analysis to put together things that share the same profile of connections and segregate the ones that have different connections. So we did that using 2 data sets. We use like the Human Connectome Project and the local data set, and we use probabilistic tractography and a very interesting method to segregate, to define the boundary between the gray matter and the white matter. You'll find the details in the paper. And we did that at the individual level. And the reason why we did that at the individual level is because you cannot use an atlas to build an atlas. This will be a circular way of thinking about saying. So we had to identify brain region at the individual level and then bring it in the stereotaxic space and then compare them across subject to assess whether or not we have the same result or different results. Basically, the way we divide and we create our region to see the tractography is through the intersection between the gray matter and the white matter that we multiply by a grid of numbers, and that gives us those very nice division of the left and the right hemisphere, completely unbiased at each individual level. Tractography, as I said, is not perfect. It has true problems mainly because of the resolution and problem of signal. So we kind of lose streamline as we're going further. So we have a distance effect. We have problem of specificity, particularly when tract tends to funnel together when they come out back. Then it's difficult for tractography to be sure it's on the right path. And the assessment of strength of connections is always a problem because the more crossings you have, the less streamline you will have, and this can bias the measure. We propose a set of solutions that are not the best, but they kind of compensate a little bit for this problem. So the distance effect is sort of covered by a multiplication by the distance, but this is assuming that there is a linear relationship between the two. Long story short, this is what we did. For the spatial specificity, we use spatial region of interest, and typically the funneling is a problem that concerns the corpus callosum projection on the contralateral hemisphere. In order to cover for that, we have a region for the corpus callosum, and information from the other hemisphere will not infer the parcellation, just the connection to the corpus callosum. And for the strength of connection, we actually normalize the pattern of connection of each region in the cortex with a z-score to have a pattern rather than a pure measure of the strength. So we know how many standard deviation above the average this region is connected with another region. Okay, you have an example of those regions at the top of the image. A is actually a region in the occipital lobe, B is a region in the frontal lobe, and C is indicating use a probabilistic tractography between the two. We ran the principal component analysis, as I was telling you just before, and found systematically division between principal and residual components that was centered around 12, suggesting that 12 regions exist in the brain. So on the left, you have a typical subject. On the right, you have all the subject together. Now, you can see that the division of the frontal lobe that we have based on the principal component shows some clear regions that remind you like important functional fields, such as on the medial wall division between the ASMA and the pre-SMA. It looks like you have the hand motor area. In dark blue, you have the frontal field. You have the Broca areas that might be here in cyan at the bottom. It is suggesting there is a link between those region and functional field. But first, how reproducible is this information? Well, we did compare the left and the right hemisphere, and if you can see, like they have very similar division with difference that are related to asymmetrical distributions of those functional fields across the hemisphere, at least putatively. We also checked whether the pattern of connection was different across those 12 regions. For the purpose of clarity, 1 represents the most posterior region and 12 the most anterior regions. You can see that the pattern of color here indicating the strength of the connection is very different across the 12 regions, whether this is within the frontal lobe or between the frontal lobe and the limbic lobe. between the frontal lobe and subcortical region, the frontal lobe and parietal lobe, frontal lobe and temporal lobe, and frontal lobe and occipital lobe. Then we assess whether this finding might be reproducible in a different data set. As I mentioned before, we use a Parisian data set to compare it with the Human Connectome project. And while it's a much more modest sequence, we've been able to see that on the left in A's, the average results that I showed to you for the Human Connectome project analysis is very comparable with our local acquisition at the bottom here, except for the dark green area, which is bigger. You know, it's a French brain. It might be the cheese area or something. And then on the right, you have the representation of individual subject parcellation based on their connections. And you can see that they all look like the model, with some differences that might well be specific to their individual organization of the brain. Now, okay, we spoke about dividing the brain with connection, but our real purpose is to see whether there is a link with functional field, as these three main authors have been saying. Let's try to have a look at the frontal field. If you compare the frontal field activations with the dark blue areas that I was speaking about, you can see that you have a good match. And then if you compare it at the individual level, you know that the frontal field is at the intersection between the superior frontal sulcus and the precentral sulcus. You can see that our region at the individual level is always located in this area. And then what we do, we expand this to any kind of functions. And you can see that each of the area I've been reporting to you before on the left show a functional specificity. You see this line of statistical significance, which is making a diagonal with very dark color on the side of it. And I mean, it is correlated with this function and not the other functions that are activated for neighboring areas. There is also a spatial correspondence as a part on the right, but I'll skip this for the purpose of time. Now, there are some very famous models of how the frontal lobe function, and one of them is a branching that is showing how, as you're going further into the frontal lobe, you have more and more advanced manipulation of the information that will contribute to making the right decision. And so we tried to see whether we could see a pattern like this in the frontal lobe as we're going further in the frontal lobe, whether we'll see changes in the pattern, in the specific information in the regions. Now, if you look at cortical thickness, you see that the further you're going toward the frontal pole, the thicker your cortex will be. And this is highly reproducible in the left and the right hemisphere and also in individual subjects. Although we were excited at first, we quickly discovered that cortical thickness derived from structural imaging is not really good. And this is mostly because the lower layer of the cortex are highly myelinated, which might bias a lot the estimate of the true cortical thickness. And you have an example here of the cortical thickness according to layers, and you can see if you forget the first layers, this will have definitely an impact on your pattern. Next, we say, well, if myelin is important, how about we look at the myelination using a T1-T2 ratio? And we saw that the further we're going toward the frontal pole, the less myelinated the cortex will be according to this measure, which would mean that the further you go, the slower the conduction inside the cortex will be. So there must be something very special in this cortex happening so that it makes us able to do all these fantastic advanced function if that's not faster conduction. We looked at the big brain, which is a fantastic resource which allows you to peer really inside the cortex at the microscopic resolution. And those slides are cell stain, and you're able to identify really and see like the soma of neurons. And so we look at how much the brain has been stained, and we've been able to see that as you're going further toward the frontal pole, actually less visually in amount of staining are stained as you're going toward the frontal pole, which will mean you actually might have less neurons in the frontal pole that you might expect. And this has been recently replicated in macaque in a paper published in eLife in 2018, where you can see that the neural density is decreasing as you're going towards the frontal pole. So, you know, it's really something that exists. So like less neurons, less myelin, more complex function, how does it work? So we tried to take advantage of diffusion weighted imaging and use this information to quantify how much water molecule diffuse inside the cortex. So we have a quantification of what we call the mean diffusivity to see really what's happening in terms of the diffusion in there. So there might be more connection and the water is going to diffuse more. There might be also more space between the neuron and the water will diffuse more. But that would give us a little information about the structure. And what we can see is, yeah, it's the same again. As you're going towards the frontal pole, you increase the amount of diffusion. There is more water molecule diffusing there, suggesting that there might be more connection, intra-connection inside the cortex there. So we thought like, okay, but how can we assess really intracortical connections? And so we thought like there must be a way to derive this information from functional connectivity. So we use resting state functional MRI and quantify the complexity of the signal in the 12 regions at rest to assess to assess intravoxels synaptic complexity. So we use an entropy method, and the way entropy works, it assesses complexity. To give you an example or a metaphor, if you use a flute and the flute has a given number of notes that will correspond to a given number of connections, you'll have a reduced number of states you can take or melodies you can take you can make with your flute, and your entropy measurement will be here 0.66. On the other hand, if you use a big flute with a lot of different notes, the number of melodies you can do is way more, and you can do like way more different states, and your entropy measurement will be 1.67 in that case. We did exactly the same thing for every voxel of the brain, except that was not flute that we were thinking about, is the number of synapses, and synapses allow you to do different notes, and the melody is the different states that your voxel can take. And by doing so, we've been able to demonstrate that the further you go inside the frontal pole, the more melodies you can do, and the more connections you might have as you have this increase of entropies that happen as you're going toward the frontal pole. So in sum, the take-home message here to take will be that there is a dorsal ventral trend in the parietal frontal cortex going from spatial to non-spatial function. And you can, so far as we know, divide your frontal lobe in a ventral part, which will be non-spatial, and dorsal part, which will be spatial. And there is also an anterior-posterior trend of the frontal lobe going from the simplest to the most advanced function, which you would have known before if you're interested in the frontal lobe. What we've been able to demonstrate is this trend is characterized by anatomical signatures such as the structural connectivity, the cortical thickness, the myelin density, the water diffusion, the intensity of silver staining that I showed to you in the big brain, and the broad signal complexity at rest, which we think it's a surrogate of the synaptic complexity